Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of the year 2019. I will be discussing the genetics section. If you are new to my channel, please do like, share and subscribe. Then coming to December 2019, shift 1. In summer squash, white color fruit is dominant over yellow color and disc-shaped phenotype is dominant over sphere-shaped phenotype. Determine the genotype of the parents if the cross between white sphere crossed with white sphere gives 3/4 white sphere and 1/4 yellow sphere. So they have already told that white sphere crossed with white sphere. So sphere is naturally a recessive character and white is a dominant character. So the parents will be capital W and small d will be present in the recessive condition because it is sphere they have mentioned, which is a recessive character. So the answer here will be option 2. That is W, capital W, small d into capital W, small d. But if you see the other options, you will see that it is capital D, which is not correct. Here also D is dominant, so that is not correct. Here also D is dominant, which is not correct. So answer here is option 2. And option 2, if you cross, you will get 3/4 white sphere and 1/4 will be yellow sphere. Then next one. Interacting genes which are involved in producing continuous variation in phenotypes in a population are known as quantitative trait loci or QTLs. Then next one. In an organism, A plus governs gray body color, while its mutant small a gives yellow body color. Further, presence of B plus allele gives long and thin hairs, while B allele gives rise to short and thick hairs. The alleles A plus B plus are dominant over A and B respectively. An individual with the genotype, that is this genotype, has a patch of yellow cells with short and thick hairs. Which one of the following event is most likely to have led to the above? Non-disjunction of the homologous chromosome during mitosis. That is not correct. Somatic recombination involving A and B. That is the correct answer. Why? Because it is not in the progeny that this condition is found. It is found in the individual. So it is somatic recombination. So answer here is option 2. Coming to the next question. The individual considered in this question are having 2 haploid set of autosomes and no Y chromosomes. The X:A ratio of the individual, the type of the organism chosen, their primary sex and number of bar bodies expected in their cells are shown in the table below. Select the options below with all correct matches. So if you see the X/A ratio of Drosophila, X/A is number of sex chromosomes by number of autosome. For Drosophila, if you see if it is greater than 1, it is super female or meta female. And 1 means female, 1 to 0.5 is intersex and 0.5 is male and less than 0.5 is super male. In case of humans, 1 is female, 0.5 is male and 2 is female. So, coming to Drosophila first. Drosophila 0.5. That is 0.5. Drosophila is a male and number of bar bodies is 0. There are no bar bodies in Drosophila. 2. Drosophila is a meta female and number of bar bodies is 0. So 1B, 1A, 2B, 3A. That is correct. Now coming to humans. 0.5 human is a female. It is not a male because if you choose male option, then option 2 Drosophila is not matching. So we have to choose 0.5A human is a female because in humans there is X0 condition and it is found in females. And the number of bar bodies will be 0. Now if you choose the second condition, that is 2 in humans, it is a meta female, it is a female and number of bar bodies is 3. 2 is when you have 4 X chromosomes. 3 will become bar body and only 1 will be functional. So it is a it is a female. So the answer here is option 1. Now next one. In the following pedigree, 3 STR loci A, B, C are linked on the long arm of the X chromosome in the order centromere, A, B, C, telomere. Further in the table, the STR alleles present in each individual is indicated. Based on the above, X chromosomes in which of the following individuals are recombinant? Hint: X chromosome in males will help identify the phase of the allele. So one basic thing everybody knows is that in females, one X chromosome comes from the mother and the another X chromosome comes from the father. Whereas in males, the X chromosome comes only from the mother. Now if you see the condition over here, that is 1, 2. 1, 2 is 4, 9, 2. So 4, 9, 2 I have written over here. 1, 4 is 4, 7, 3. 4, 7, 3. Now 2, 1. 2, 1 is you can see 4, 9, 2 and 6, 7, 1. 4, 9, 2, 6, 7, 1. So this 6, 7, 1 must have got from the female parent. 6, 7, 1 over here. 6, 7, 1. 1, 1 is 6, 7, 1. And 4, 9, 2 is the male parent. So this is a female child. So she will inherit X chromosome from mother and X chromosome from father. There is no recombination there. So 1, 2, 1 is not a recombinant. Then this is a male. So 6, 8, 1. 2, 2. This one. 6, 8, 1. 6, 8, 1 is a male. That is, there is also no recombination. Then third one, if you see, 6, 8, 1 and 6, 9, 2. So 6, 8, 1. These are the parents. 6, 8, 1 got from the father and 6, 9, 2 obtained by recombination between 4, 9, 2 and 6, 7, 1 in the mother. So 3, 1 is a recombinant. Now 4, 9, 2. 4, 9, 2 obtained from the mother only. So that is a male child. So 4, 9, 2. Now 3 is also a male child. So 6, 7, 2. 6, 7, 2 obtained by recombination between 4, 9, 2 and 6, 7, 1. So 6, 7, 2 is a recombinant. So 3, 3 is also a recombinant. And 4 is a female. It is 6, 7, 1 and 6, 8, 1. They obtained both from both the parents without recombination. So they have asked for, based on the above X chromosome, in which of the following individuals are recombinant. So say 3, 1 and 3, 3 are recombinants. So the answer here is option 3. Coming to the next question. A mutant mating type MT strain of Chlamydomonas that was resistant to the antibiotic kanamycin and herbicide PPT was crossed to a wild type mating MT plus kanamycin sensitive and PPT sensitive strain. 20 tetrads of the progeny were analyzed for mating type and resistance, sensitivity to kanamycin and PPT. The following observations were made. Now if you see the tabular column, you will come to know all are kanamycin resistant. That means kanamycin resistance is inherited maternally. So option C, Inheritance of kanamycin resistance or sensitivity is demonstrating nuclear resistance is not correct. C is not correct. We will see the other options. But before coming to that, if you see over here, you can see that all these are parental mating types. MT, PPTR are parental, MT, PPTR again parental, MT plus PPTS, MT plus PPTS, it is parental. And this type 2 all are recombinants. MT, PPTS is recombinant, MT, PPTS is recombinant, MT plus PPTR, MT plus PPTR is recombinant. Third type, if you see, MT-PPTR is parental, MT-PPTS is recombinant, MT plus PPTR is recombinant, MT plus PPTS is parental. Now the number of each type observed. So there are 8 here, there are 4 tetrads. So 8 into 4 will be 32, 9 here, 9 into 4 will be 36, 3 into 4 will be 12. Now, so the total number of cells will be 32 plus 36 plus 12, that is 80. And percentage of recombinants, number of recombinants by total progeny into 100. Recombinants, I told you, it is the type 2 one, all are recombinants. So 36 plus, and in type 3, only 2 are recombinants. And there are 3 into 4 we did, that is coming out to be 12. So out of 12, that is total cells in type 3 tetrad is 12. Total cells in type 3 tetrad is 12. So out of 12, 2 are recombinants. So that will be 6. So 36 plus 6 divided by total progeny is 80 into 100 will be greater than 50%. So it is greater than 50% means linkage is absent. So if you see option A, MT and PPT loci are on 2 different chromosomes. So that is correct. Then B, inheritance of mating type and PPT resistance or sensitivity are demonstrating cytoplasmic inheritance. It is not PPT that is demonstrating cytoplasmic resistance. It is kanamycin that is showing cytoplasmic cytoplasmic inheritance. Then statement 4. Nuclear inheritance is being demonstrated by mating type and PPT resistance or sensitivity analysis. That is correct. So option 2, that is A and D, both are correct. Then next one. The pedigree shown below depicts that the individual 1 is heterozygous for dominant disease allele D and for the molecular markers M1 and M2. The paternal molecular markers present in the progeny individual are indicated in the pedigree. The following statements can be made from the above pedigree. Now we will start from C. If load score comes out to be 3, then it ensures that the 2 loci are independently assorting. Now load score is logs of the odds. So if load score is positive, it means linkage, and if load score is negative or less than 1, it is unlinked. So option C, it is said that load score is 3 and it is independently assorting, which means there is no linkage. So statement is wrong. Then option statement D. Load score is less than 1. That means it is negative value. Would have ensured that the 2 loci are linked. It says that the 2 loci are linked and they are having a negative value for load score. The statement is wrong, as if load score is negative, it means they are unlinked, but they are telling it is linked. So in the option wherever C and D comes, they are wrong. So 1 is wrong, 2 is wrong, and 4 is also wrong. So the correct answer will be A and B. Answer is option 3. The next one. Given below are various types of molecular markers in column A and properties of these markers in column B. So RFLP. RFLP is single locus. SSR is multi-locus. B2. C. AFLP is dominant. Multi-locus. And RAPD is dominant. So the answer here is option 3, that is A1, B2, C5 and D4, D6.